Hello everyone, welcome back to my channel. It's your favorite biology teacher, Najuma Sharuk. I am here to take you through microscopy. Under this section, we are going to cover how a light microscope is used. We are going to look at the difference between an electron and a light microscope. We're going to look at the difference between magnification and resolution. We are going to look at the reason as to why light microscopes have a limit on how much detail we can see when we are looking at a cell. So in this section, all we are going to cover about is microscopy. Let's get started. In our previous lesson, we were able to see that microscopes magnify cells. That's why we are able to see cells, and we only have two types of microscopes. We have the light microscope and the electron microscope. A light microscope is the one you're going to find in your lab, the one that you'll use in your AS practicals. The electron one, you might not really find it in your labs, or it's not so common because of the fact that it's really expensive. You can imagine maybe it starts from roughly, if you look at the price, it starts from roughly $50,000 and above. And many schools cannot really afford to buy such a gadget. You mainly find it in universities. Yes, like here in Uganda, you find it at Makerere University. Actually, the only electron microscope in Uganda is at Makerere University, you can imagine. Okay, so remember that when you're to observe an object under a light microscope, the light microscope uses light as a source of radiation. Radiation is the type of energy that type of microscope is going to be using. So the light microscope uses light energy. And how does this microscope work? We are going to have a section where the specimen is placed. Assuming the specimen is placed here, usually there is a stage where you place the slide that has the specimen, and light is supposed to pass through that specimen. Now, when light passes through the specimen, there are lenses. There is an objective lens which magnifies the object, and then there is also an eyepiece lens which magnifies the object. Then you place your eye here so that you're able to observe the magnified specimen. Now, your eyes can be able to interpret light. That is why you can use your naked eye to see a light microscope, because our eyes are able to interpret light in terms of images. So our brain can interpret light and make images. And that is why when you're using a light microscope, you can directly observe the specimen using your own eyes. And when it comes to this light microscope, it uses lights. However, the electron microscope uses electrons. That is why it is called an electron microscope. So in case you are using a very high quality light microscope, this is what you will see under a very good quality light microscope. If you're observing an animal cell, these are the different structures that you're going to see. If you're observing a plant cell, these are the different structures still that you're going to be able to observe or see. Now, when it comes to animal cells, you can see that we can be able to observe the mitochondria, the cell surface membrane, the nucleus, the chromatin, DNA inside the nucleus, the nucleolus, a centriole, Golgi body, cytoplasm. But we have these small structures. They are telling us these small structures are difficult to identify. That means that however much of a very high quality microscope you're using, even if that microscope is of very good quality, there are some structures inside a cell that you'll not be able to identify. Like you'll not be able to see their details. It's the same case for a plant cell. These are the features you'll be able to see in a plant cell if you're using a high quality microscope. But when it comes to this plant cell, still there are going to be some features here. You can see they are labeling these small structures that are difficult to identify, which simply means that even if the microscope is very good, as long as it's a light microscope, there is a limit on how much detail you're going to be able to see. When you look at these two plants and animal cells, you should be able to compare them. Look at the similarities, look at the differences. Because such simple questions can be in your paper where they tell you to compare plants and animal cells. Plant cells have some features which are similar to those found in animal cells. And at the same time, plant cells, anyway, generally, when you look at plant cells, they have more features. You can see they have a cell wall, which you will not find this side. They have chloroplasts, which you will not find in an animal cell. So you should be able to describe those features. And one other thing is that you should be able to know which features will I be able to observe in a cell if I'm using a light microscope. And then there are other features that you'll never be able to see. Why? Because those features are too small. The light microscope cannot be able to show them to you. You will not be able to see them using a light microscope. These are diagrams from microscopes, and they're from a light microscope. So these are cheek cells from a light microscope. Remember, I told you that a diagram or a photograph from a light microscope is called a photomicrograph. So this is a photomicrograph of human cheek cells. Hope you can see the cell surface membrane, the nucleus, the cytoplasm. Then even here we have the moss leaf cells. You can see the cell wall, the chloroplast. This is from a plant cell. This is from an animal cell. So when you look at a cell, many of the contents in a cell are colorless. When you look at how a microscope works, a microscope works in a way that if this is the specimen to be seen, observed, when light is trying to pass through that specimen, the structures inside this cell have to be able to obstruct that light. If they obstruct the light, then an image can be made. I can give you an example of a shadow. You can only form a shadow if you obstruct light. If you stand in the sun and you obstruct light, then a shadow will be formed. Your shadow will be formed. So take the shadow to be an image. When it comes to the light microscope, for specimen to be seen, for different structures to be seen, those structures should be able to interfere with the light waves, or they should be able to obstruct light. So it's only those features that can obstruct the light waves that their images are going to be observed. Now, one of the challenges is that most of the cell contents are colorless. So if the cell contents are colorless, that means that when light is made to pass through those cell contents, that light just passes through. In anything that is colorless will just allow light to pass through because it will be transparent. Light will pass through and an image will not be made. So for these cell structures to be seen, since they are colorless, for them to be seen, they have to be stained. If we go back to our previous slide, which was showing us the human cheek cells, like an example, when you look at these cells, you can see they have some color. Now, this color is not the color that these cells have. All the cells and their contents are colorless. So usually, special stains are used. An example, you can see this nucleus is stained, and it means that a special stain was applied to stain this nucleus. That is why it was visible through that microscope. Even the cytoplasm has also been stained. You can see that you can now see the cell membrane because it has been stained. So when staining specimen for viewing in a light microscope, special dyes are used, colored dyes. So colored dyes are used for staining in light microscopy. If you're observing images using a light microscope, you're going to use colored dyes to stain. When it comes to the plant cell, like this one, since it already has chlorophyll, chlorophyll is a green pigment. So the chloroplasts, you might not need staining because these chloroplasts will already be visible since they are colored. They'll be able to obstruct the light and then you'll be able to observe them. So I'm posing a question for you, basing on the structure of an animal and a plant cell on the previous slides that we have observed. Are you able to tell features that are common or structures that are common between animal and plant cells? Are you able to tell features that are found only in plant cells? Are you able to tell features that are found only in animal cells? You should be able to do that. If you can't, go back. You can take the video back and check those features so that you're able to tell which ones are the same in both plants and animal cells, which ones are only found in plant cells, or which ones are only found in animal cells. So features that are found in both plants and animal cells: cell surface membrane, a nucleus, cytoplasm, organelles such as mitochondria and Golgi apparatus. Yes, if you look at the mitochondria, it's this side also. Cell membrane, it's also found this side. Cytoplasm is also found this side. Golgi body is also found in both plants and animal cells. Let's look at an example of an exam question. Here they're asking you which structures are visible in an appropriately stained plant cell using a high power microscope with a magnification of times 400. And that is a light microscope. We have mitochondria, we have starch grains, we have centrioles. So a tick means it's visible, a cross means it's not visible. We are looking at centriole, mitochondria, starch grains. If we go back, let's try to see. If we look at this plant cell, they said chloroplasts. Yes, we shall be able to see a chloroplast if you're using a light microscope. Mitochondria will be visible. But the other third structure is a centriole. Plant cells have no centriole. Centrioles are only found in animal cells. That means that for the centriole, it will be a cross. So that means that the most appropriate answer is D. The most appropriate answer is D. So these cells are going to have organelles within them, and someone would be wondering what an organelle is. An organelle is a part inside a cell that plays a particular role. Like a mitochondrion is an organelle, and a mitochondrion even has a membrane surrounding it. A nucleus is an organelle. It plays a particular role for this cell. It even has a membrane surrounding it. A Golgi body is also an organelle. So organelles allow division of labor within a cell. What do you mean by division of labor? Different activities, different reactions will be happening in each of those individual organelles. We can look at that as compartmentalization. I can give you an example. When you go to your school, you'll find that there are different classes, year 9, year 10, year 12. All of you are in the same school, but when it comes to learning, you go to your particular class. That means that the people in year 9 will be able to study without any interruption from those in year 12. So that is compartmentalization. So even a cell shows compartmentalization by having different organelles that are responsible for carrying out different functions. Organelles have membranes surrounding them. So that is why you always find that word, membrane bound. Membrane bound simply means it's surrounded by a membrane. like a nucleus has a membrane surrounding it, so it's membrane bound. Now, we have another term that we need to learn, which is resolution. Resolution. Resolution is used in microscopy. This term is used in microscopy. So someone could be wondering, what is resolution? Let's look at an example. I have this micrograph. It's from a microscope. It's labeled B. Then I have another one labeled A. When you look at these two micrographs, one is from a light microscope, one is from an electron microscope. Which of the two is much more clear? Which of the two, is it A or B, which is much more clear? Yes, it's B. You can see that B is much more clear compared to A. And now the fact that B is much more clear compared to A, we can say that the microscope that was used to observe this structure has a higher resolution. Why? Because the image made by that microscope is much more clear. And actually, this micrograph B is coming from an electron microscope, but micrograph A is coming from a light microscope. Let us just look at this section here in both microscopes. So in both micrographs, you can see this section here and then this same section here. Now these are chloroplasts. This is a chloroplast, another chloroplast, another chloroplast. You can see that when it comes to this microscope, it was able to show you that these chloroplasts are distinct. Each is on its own individually. However, when you look at this micrograph, the microscope was showing you as if this structure is the same. But this microscope was able to show you the details that no, these are three different chloroplasts. However, here they are seen as one. So that takes us to the term resolution. So structure B or micrograph B was made from an electron microscope, whereas A comes from a light microscope. So a light microscope has less resolution compared to an electron microscope. So when someone talks about resolution, we are looking at how much detail can you see if you're using a microscope? How much detail can you see? Obviously, in this micrograph B, we are seeing much more detail compared to A. In A, you can see that some of the structures are even blurry. But in B, they are much more clear. So resolution is the amount of detail that you can see, the amount of detail that can be seen. So electron microscopes are able to show you much more detail compared to light microscopes. Resolution can also be defined as the ability to distinguish between two objects that are very close together. Let's go back to these micrographs. You can see these chloroplasts are very close together, and the microscope was able to distinguish them and show you that they are different. However, the light was not able to distinguish them. It wasn't able to distinguish them. It shows you as if it's the same, it's just one structure. However, here it's showing you that they are different, three different chloroplasts. So resolution is the ability to distinguish between two objects that are very close together. The higher the resolution of a microscope, the greater the detail that can be seen. So if an image was made with a very high resolution, then even the detail that you're going to be able to see is also going to be much more. Like from this diagram, we have much more detail, which means higher resolution. Here, the resolution is lower. You can compare that. Different people have different cameras or different phones. Obviously, the resolution of an iPhone X will not be the same as that of an iPhone 17 because there is a lot of advancement and the cameras have different resolutions. That means that the pictures will vary that are going to come out of such a camera, and that is the same case here. Or the resolution of an Infinix phone obviously will not be the same as that of an iPhone. So when it comes to the light microscope, it has a resolution of 200 nanometers. 200 nanometers. What does that mean? It means that if an object is smaller than 200 nanometers, you will never see it using a light microscope. You'll never be able to see it. It will not show it to you, which simply means that if still objects are close together and the distance between them is smaller than 200 nanometers, the light microscope will show you as if those two objects are the same thing. It will not be able to distinguish them. It will not be able to see them as separate, just like from the previous slide that we have seen. So the resolution of a light microscope is 200 nanometers. And someone will be wondering, why is it that a light microscope can only show you structures that are bigger than 200 nanometers, but those which are smaller than 200 nanometers, you'll not be able to see them. And this means that even if you try to increase the magnification, the images won't become much more clear. They will just become blurry. They will just become blurry simply because the microscope has a limit on how much detail that it can show you. So the question is, why? Why is it that there is a limit on how much detail a light microscope will show you? Why is it that its limit is 200 nanometers? So this takes us to the electromagnetic spectrum. Under the electromagnetic spectrum, we are looking at the range of different wavelengths. Here we are looking at different radiations, and these radiations are moving at different wavelengths. We have X-rays, we have gamma rays, UV rays, we have light, visible light, the light that we can see. We have infrared rays, microwaves, TV waves. You can see that these waves are of different lengths. Some are longer. And if the wavelength is longer, that means that the waves are going to be much less frequent compared to shorter wavelengths. So shorter wavelengths usually are more frequent compared to longer ones. You can see the radiant TV waves are really long. If you come back to this side, the gamma rays, the wavelength is really short. It's a short wavelength. So when it comes to visible light, what is the wavelength of visible light? Visible light has a wavelength that ranges from 400 nanometers, that is for violet light, to 700 nanometers. So visible light is the type of radiation that is used under a light microscope. So remember that for an object to be observed under a microscope, it should be able to interfere with the light waves. Let's take an example on the next slide. If you look at, let's use this diagram here. Let's use this diagram here. If you look at the light waves, we are going to use the shortest wavelength, which is violet light. Violet light has a wavelength of 400 nanometers. If you look at violet light, as that light moves, for an image to be made, the structure inside a cell or inside a specimen that is being viewed has to interfere with the light waves. And in this case, we are having two structures. We have a mitochondrion and we also have a ribosome. Mitochondria have usually a diameter of around 1000 nanometers, so they are big in size. You can see they are bigger than 200 nanometers, which is the limit of resolution by a light microscope. So when you use violet light, you can see that the mitochondrion is large and it's able to interfere with these light waves. This means that you can be able to observe this mitochondrion. But when it comes to the ribosomes are too small. When the light waves are moving, they just jump over them. That is why you'll never be able to see ribosomes if you're using light. Because even if we use the shortest wavelength, which is 400 nanometers, still the ribosomes cannot interfere with those light waves. And when you look at the ribosomes are 25 nanometers. 25, which is way much smaller. 25 is way much smaller than the 200 nanometers, which is the limit of resolution by a light microscope. So that is why some structures cannot be seen using a light microscope, simply because they are too small and they cannot interfere with the light waves. So you could be asked a question, why is it that ribosomes can never be seen? Explain why ribosomes are not visible using a light microscope. Mention about their size. Ribosomes are 25 nanometers. The light microscope has a maximum resolution of 200 nanometers, which is way bigger than 25 nanometers, which simply means that you cannot see the ribosomes. Why? The ribosomes will not interfere with the light waves. They will not interfere with the light waves. So that is why you cannot see the ribosomes using a light microscope. So because of the fact that the light microscope has a limit on how much detail can be seen, anything smaller than 200 nanometers could not be seen using a light microscope. That is why they had to look for other solutions. They had to see more, they wanted to see more detail. They were like, so what do we use? Light has a limit. So what next? And they had to think of maybe using another type of radiation. If you look at this electromagnetic spectrum, we have radiation. This is visible light, which had a limit on the resolution. And then we have these radiations this side and radiations this other side. So when you look at this electromagnetic spectrum, the possible radiations that they could have thought about using were the ones this side, which have a shorter wavelength, shorter than 400 nanometers, a shorter wavelength. So they tried using UV rays, but the UV rays could not easily be focused. Even X-rays could not easily be focused. They could not be focused. Remember, if the wavelength is short, that means that if an organelle is small, assuming this is our organelle, for a short wavelength, it will be able to interfere with that wavelength, and then an image will be made. But if the wavelength is long, it will just pass over the organelle. The organelle will not interfere with it, so an image will not be made. So that is why they had to think of shorter wavelengths. So they couldn't use the X-rays, they were difficult to focus. Even the X-rays, UV rays, gamma rays were difficult to focus. So that is how they thought about using electrons. So the much better solution was to use electrons. Why? Number one, electrons are negatively charged. We know that. And when a metal gets excited, electrons move to higher energy levels. They gain so much energy and they escape their orbits. And when they have escaped their orbits, they move like a radiation. That means they move in waves. They move in a wave. They move in a wave. And the beauty about the wavelength of these electrons, it was realized that their wavelength is really very short. So that is why they were very good candidates for microscopy. You could find a question asking you, why is it that electrons were a very suitable form of radiation for microscopy? The first answer that should come to your mind is that their wavelength was extremely short. And then because they are negatively charged, they could be focused. They could be focused using electromagnets. So a light microscope uses lenses, glass lenses to focus light, whereas an electron microscope uses electromagnetic lenses to focus electrons. So using an electron microscope, a resolution of as low as 0.5 nanometers was obtained. Remember, a light microscope, its resolution was 200 nanometers. But here we are with an electron microscope with a resolution of just 0.5. If you do the math, you're going to find that the electron microscope was 400 times better than a light microscope. This means that even ribosomes, remember ribosomes were just 25 nanometers. But then the resolution of an electron microscope is 0.5 nanometers. That means that ribosomes are still bigger and they are going to be observed using an electron microscope. So that is how an electron microscope came into play. And it has really been very important. It allowed scientists to be able to see much more details that are found within a cell. So there are two types of microscopes, of electron microscopes. And the first one is what we call a transmission electron microscope. A transmission electron microscope. The second one is a scanning electron microscope. So you will usually find such terms as TEM. TEM standing for transmission electron microscope. Or you'll find a transmission electron micrograph, which simply means a photograph that was taken from a transmission electron microscope. Like this is a transmission electron micrograph. So how does a transmission electron microscope work? Now remember that electron microscopes use electrons. That means that if this is the specimen to be observed, electrons have to pass through the specimen. Transmission. Electrons are emitted and they pass through the specimen. So the electrons have to pass through the specimen. And usually there is a screen where those electrons will fall. Remember, you cannot see electrons. For light, you could see light. That is why you just place your eye on the eyepiece. But for the electron microscope, there is always a screen, a fluorescent screen, where those electrons have to fall. When the electrons fall on that screen, then an image is made. An image is made. So the electrons that are able to pass through fall onto a screen and an image is made. Most of the time, the image that is made is in black and white. That is why in your papers, you find a lot of diagrams that are in black and white, black and white, black and white. Yeah, so the beam of electrons is passed through the specimen before being viewed. Only those electrons that are transmitted, that is why it is called transmission, only those that are transmitted will fall on the screen and then an image is made. That means that the region where these electrons have been passed is going to be displayed on that screen. So these transmission electron microscopes allow you to see details inside thin sections of specimens. Like an example, this is from a plant cell. You can see the details inside. You can see a nucleus. Maybe this is a mitochondrion or a chloroplast. vacuoles, small vacuoles, cytoplasm. So you can see the details inside. So as long as you can see details inside a cell, just know that was a transmission electron microscope that was used. It is able to show you details inside cells. We have a question here. You could be presented with such a diagram. They are telling you outline one feature of this figure aside that confirms that the microscope used to obtain this image is a transmission electron microscope. What are some of the possible answers you will write? Outline one feature of this figure that confirms that the microscope used to obtain that image is a transmission electron microscope. Okay, feature number one, I'm able to see internal features. I can see these internal features, which simply means it's a transmission electron microscope that was used. And then it's a very thin section. It's a thin section that is being shown. And these electron microscopes, especially the transmission ones, provide you images that are in 2D. So this is a 2D image. This is a 2D image, and these show us that it is a transmission electron microscope that was used. Then how about the other type? The other type is the scanning electron microscope. It just scans the surface, only the surface. So for it, it only shows you surface features. Someone could be interested in seeing the details on the surface of an organism. Like in this case, this is a cat flea head. Now you can see the details of the surface of this head. So for it, how does it work? If this is our specimen, and then a beam of electrons comes in, that beam will not pass through the specimen. So if this is our beam of electrons, the electrons are going to be reflected. The electrons are reflected, and then they fall on a screen still, a fluorescent screen. When they are reflected, they will fall onto a screen, and then an image is going to be made on that screen. Still, this will give you black and white pictures. But usually when the pictures are made or the images are made, they are false colored using a computer, maybe to make them more appealing. Yeah, but the images most of the time are in black and white. So it's only the reflected beam that will fall on the screen and then an image is made. That's why it's called scanning, because it just shows you details of the surface. And when it comes to the scanning, its resolution is lower than that of a transmission. Its resolution lies between 3 nanometers and 20, whereas the transmission, we saw that its resolution was 0.5 nanometers. Let's look at an example of a question still about a transmission electron microscope. Sorry, a scanning electron microscope. So here we have a figure 3.1 showing us a scanning electron micrograph of a pair of human chromosomes. So yeah, these are human chromosomes. And we have a question here. Outline one feature that confirms that the microscope used to obtain this image is a scanning and not a transmission. Scanning and not a transmission. What would be your possible answers or submissions? Number one, we are able to see just surface features. You can even see the contours. Just surface features. We do not see any internal features. So no internal features are visible. We are only observing or seeing surface features. It's not a thin section. You can see this is something that is really thick. And then these scanning electron microscopes give you 3D images. Yeah, this image is in 3D. So those are the possible answers that you could have given. So how does this electron microscope work? Now, the electron microscope has a section where you place the specimen to be observed. The specimen can be placed here. Remember, all we need is electrons. So we are going to have an electron gun. The electron gun produces electrons, and those electrons are able to be focused by an electromagnetic lens. So the lens focuses those electrons so that they pass through the specimen. Now, since these electrons are going to pass through the specimen, this means that here we are looking at a transmission because the electrons will pass through the specimen. And then down here we have a screen where the electrons fall so that an image is made. So remember, the image made is going to be in black and white. Now, even specimens that are to be viewed using electron microscopes also have to be stained. We saw that in a light microscope, colored dyes are used for staining. However, in an electron microscope, heavy metals will be used for staining. Heavy metals, remember, metals can obstruct electrons. So heavy metals will be used to stain specimens that are to be viewed using an electron microscope. So at the end, an image is going to be made. Now, some important features to note about an electron microscope is the fact that these electrons have to be moving in a vacuum. So a vacuum is needed. Why is it that a vacuum is needed? Number one, if you look at electrons, if they are moving, if there were air particles, the air particles would collide with the electrons and prevent electrons from being focused. That is why there should be a vacuum so that the electrons are focused without any collision with air molecules. One other important feature to note is that water boils at room temperature in a vacuum. Imagine water boiling at just 24 degrees Celsius. If it is in a vacuum, it will start boiling at 24 degrees Celsius. 24, 25. So what does that mean? This means that if you get a specimen that has water, and remember, most cells have water. Cells have water. If you get a specimen that has water and you place it in this microscope, it means that that water is going to start boiling and evaporating. So this is why any specimen to be observed using an electron microscope has to first be dehydrated so that all the water is removed. Because if you do not dehydrate it, water will evaporate and then interact with this beam of electrons. And then the electrons will not be focused, thus you'll not have an image made. So specimens have to be dehydrated so that we have no water. And that is why, and remember, when you dehydrate, you kill. That is why these electron microscopes only use dead material. They only use dead material. They will not be using live material. Because the moment you dehydrate a specimen, it means you're going to be killing it because you have removed all the water from it. So those are the major differences between an electron microscope and a light microscope. You should be able to tell the differences between an electron microscope and a light microscope. An electron microscope and a light microscope. In summary, we have seen an electron microscope uses electrons as a radiation. A light microscope uses light as a radiation. An electron microscope works in a vacuum. A light microscope doesn't need a vacuum. An electron microscope uses electromagnetic lenses to focus the electrons. A light microscope uses glass lenses to focus light. An electron microscope uses dead material. A light microscope can use both dead and live material. An electron microscope gives you images in black and white. A light microscope gives you images that are colored because you can interpret color. A light microscope, or which other one did I want to mention? Any, those are the major differences between a light microscope and an electron microscope. So we can call it a day for today. See you again on the next one. Thanks for being around.